Okay, 2 minutes past. Good morning and good afternoon, ladies and gentlemen. Happy Friday and a warm welcome for the latest installment of the CDX Engage webinar series hosted by Angle. This one is entitled The Value of Incorporating Circulating Tumour Cells into Clinical Trials, a Paradigm Shift. My name is James and I'm the CDX Engage team here at Hanson Wade, and I'll be chairing the session with you today. So the session is going to focus on how pharmaceutical companies can best unlock the value of CTCs for clinical trials. We'll first be hearing from Evie Lianadao, the Professor of Analytical Chemistry and Clinical Chemistry at the University of Athens. She'll be sharing the importance of looking beyond the purely epithelial CTCs, articulating the depth of insight that can be gained looking at cancer from pushing beyond just CTC enumeration and looking at protein and genetic markers. We'll then be hearing from Madeleine Repellet, the head of clinical laboratories at Angle. With her 30 years of clinical laboratory experience, the most recent 21 of these working for liquid biopsies, you can ensure that there is no one better to share thoughts on the practicalities of integrating CTC analysis into clinical trials. So we plan to finish in just under an hour, and we'll be packing in 2 fantastic presentations followed by a Q&A where you'll be able to have your questions answered. So please note the questions tab at the bottom of the webinar dashboard, where this is where you can submit your questions. So without further ado, we begin the webinar with Dr. Lianadao. Her molecular diagnostic laboratory specializes on CTC analysis. Dr. Leonardo has 132 publications and is not new to presenting her work, having organized 4 international liquid biopsy meetings. From 2014 to 2019, she was chair of the Committee for Clinical Molecular Biology Curriculum for the International Federation of Clinical Chemistry, the IFCC, and in 2020 became an elected member of the IFCC Committee on Proficiency Testing. So I'd now like to wish Evie to join me on the stage, and we begin the presentation of the value of incorporating CTCs into clinical trials. Okay, hi Evie, I can now see you in the presentation.
Okay.
Thank you.
Thank you.
Fantastic. I'll leave it over to you.
Okay, thank you for the invitation. And now I will start showing, I will start presenting my talk. First of all, I would like to thank Angle for this invitation. And my talk will be on the value of incorporating CTCs into clinical trials. This is the outline of my talk. I will try to focus on the importance of CTC beyond enumeration, the importance of VMT. I will talk about some important biomarkers revealed through CTC molecular characterization. I will go on with a direct comparison of CTCs and ctDNA. I will discuss a case study as well as the importance of CTC assay standardization. First of all, liquid biopsy is coming to fill some clinical needs in oncology, like early cancer diagnosis, prognosis, residual disease, risk of relapse, treatment selection, monitoring disease burden, but most importantly, pharma can get a wealth of information at regular intervals during clinical trials through CTC analysis. Liquid biopsy can offer a lot of information, and beyond ctDNA, methylation, circulating microRNAs, and extracellular vesicles, CTC analysis is highly important because it can give information on protein expression, gene expression. We can do functional studies, single cell analysis, and we can also check for tumor heterogeneity. So we will focus now on CTCs. Now, CTC enumeration has started many years ago and has led, starting from this seminal paper by Massimo Cristofanilli already in 2004, has led to the FDA clearance of the cell search system for CTC enumeration. That was followed also for metastatic prostate cancer and metastatic colorectal cancer. But what is beyond epithelial CTCs? And now we can talk a little bit about epithelial mesenchymal transition, which is highly important for CTC analysis and for metastasis in general. So EMT is highly important because it can affect the immunobiology of carcinomas, can cause enhanced migratory capacity in cells, increase invasiveness, and offer elevated resistance to apoptosis. And this is through cell-to-cell extracellular matrix interactions that are all are leading cancer cells to have increased metastatic potential. You can see here that by going from epithelial to mesenchymal type through EMT, the invasiveness of cancer cells is increased, as well as drug resistance and tumor initiating ability. And here you can see a number of genes that play a crucial role in EMT, which is very well studied nowadays. And I would like to point out here twist, snail, and slug as some, and vimentin as some of the most important ones. The important thing is that through these very elaborate studies of the group of Jean-Paul Thierry, you can see that the EMT score is different between different types of cancer. You can see here, for example, that colorectal cancer is mostly epithelial, while in lung cancer we have an increased EMT profile. And the same exactly picture is seen in the relevant cancer cell lines. And in real experiments, it has been shown that CTCs are actually a hybrid between epithelial and mesenchymal states.
Evie, sorry to interrupt. I can't see the screen. You're sharing your PowerPoint, but it doesn't seem to be changing slides. I just wanted to check that you're changing your slides at the moment.
Yes, of course.
Okay, I'm still seeing the value of incorporating your opening slide page, and I'm just wondering if we could try. I don't know
why. Don't you see my slides?
I see your home slide, I believe, but not the other ones. So I'm just going to try turning that off and on and trying that again if possible.
Okay, thank you.
Great, okay.
Can you see my screen now?
I could see it before. If you just escape full screen.
You mean this?
Yep, and then try again.
So, you mean show screen? You want me to show my screen?
Yes.
Okay.
Okay, great. And then if you full screen on there.
So?
Yep, and then just go to the next page. Yes, okay. Sorry, please carry on where you were. Sorry, it just wasn't changing, so please click through.
So I'm going to where I was before. Yes, I'm not starting from the beginning.
I think perhaps on the pages where you start pointing out where the graphs are, that's probably a good point to go to. Apologies about that, everyone.
Okay, here is the slide showing CTCs and the importance of CTCs. You should have told me earlier. I'm really sorry why this happened. It's not my fault. I just pushed picked the button show screen as okay. Now it's CTC enumeration. I was showing these slides that are well known to everybody on the FDA clearance for CTC enumeration in metastatic breast and prostate cancer. I was talking about epithelial mesenchymal transition, or EMT, saying that it's highly important because the invasiveness and drug resistance and tumor initiating ability is increased in CTCs when they are going through the EMT program. Here are some major genes that are important for EMT, pointing out mainly twist1, snail, slug, and vimentin. And here are the EMT spectrum of cell lines and tumors, and it's highly important to see that while colorectal cancer seems to be mainly epithelial, a lung cancer has more an EMT profile. And the same picture is seen in corresponding cancer cell lines. This is a slide by Jean-Paul Thierry presented at the 4ACTC meeting in Corfu last year. And here you can see that CTCs can go through epithelial and mesenchymal state, and in between we can have CTC that are mostly epithelial or mostly mesenchymal, and this is mostly can be seen when we have CTC clusters. Again, in breast cancer, you can see that we can have different epithelial mesenchymal transition states between different subgroups. For example, triple negative breast cancer is getting to be more EMT type. And in this nice work by Galatia Kalergi in the University of Patras, and that performed the university when she was in University of Crete, you can see the same different CTCs from the same patient undergoing EMT. This one is overexpressing twist1 and CK at the same time, while this one is overexpressing CK, vimentin, and twist1 in the same time. These are confocal microscopes photos. In this clinical study, it was shown in early breast cancer, it was shown that EMT was detected in almost 18% of patients, and that CTCs with an EMT phenotype had a prognostic value in primary breast cancer. And this prognostic value was demonstrated in all subgroups of patients. Our recent study in CTCs again have shown in early breast cancer, using multiplex PCR and gene expression, that twist1 overexpression was highly prognostic in these patients. This means in early breast cancer patients after surgery and before getting chemotherapy. Twist1 overexpression in this CTC is highly prognostic. We have also shown in another study in high-risk prostate cancer patients that EMT markers are highly increased after therapy in patients, while epithelial markers are almost disappearing. Epithelial markers are highly expressed before therapy, and these are the same group for the same 27 patients before and after therapy. There are numbers of CTC with low EpCAM expression, and there was a study performed in 50 cancer cell lines some years ago, 10 years ago, showing that 20% of these cell lines had very low levels of EpCAM. And these CTC will be very difficult to be captured by EpCAM. This is the Parsortrix, one of the size-based isolation systems for CTCs, which is EMT independent. Using this technology, we have recently shown in head and neck cancer that by a direct comparison study using EpCAM beads and Parsortrix and doing downstream RT-qPCR analysis, multiplex RT-qPCR analysis, that size-based isolation of CTC has a lot more positive events in the same patients, showing that CTC positivity was highly increased, both for gene expression and DNA methylation. Now going on molecular characterization of CTC. Using CTC, we can get information not only on DNA, like ctDNA analysis, but we can also get additional information on proteins based on different technologies, on RNA, and at the same time we can perform single cell analysis, which is highly important for tumor heterogeneity. Now we'll focus on some important biomarkers in 3 different types of cancer: breast, prostate, and lung. In breast cancer, I will show you some examples on HER2, PI3 kinase, ESR1, and PD-L1. And these are all connected with specific treatments, as you all know. Now, it has been shown that in CTC, some therapeutic targets like HER2 are highly discordant between primary tumor and CTCs. And this has a lot of clinical implications, as you can imagine. PD-L1 immunotherapy has also been shown that PD-L1 is present in CTC, and that CTC are highly discordant and highly heterogeneous even in the same patient. In this beautiful study, it has been shown that PD-L1 is frequently expressed in CTCs, more than 60% of patients with metastatic breast cancer, while in the primary tumor, PD-L1 was not shown to be overexpressed. And this is now checked in a high number of patients in specific clinical trials. Concerning PI3 kinase mutations, these have been also studied in CTCs, and to do this, highly sensitive mutation technologies should be developed. One of these is the one that we developed in our lab, and the sensitivity is 0.05%. And using this technology, that's a method based on real-time PCR and ARMS PCR and high-resolution melting, we have shown a 20% CTC positive for PI3 kinase mutation in early breast cancer and about 35% in metastatic breast cancer. Concerning ESR1, it has been shown that ER expression is different in sometimes in CTC than in primary tumors, and that there are ER1 negative CTCs in circulating in patients with ER positive tumors, and these cells may survive endocrine therapy. This was already shown 7 years ago. Going on with ESR1 gene, a number of mutations on this gene have been shown, and these mutations are clearly connected now with the presence of resistance to endocrine therapy. Moreover, ER epigenetic alterations on estrogen receptor driving this, the silencing of this gene have been shown for the first time by my group in clinical cancer research 2 years ago. And we've actually seen that in CTCs isolated through EPCAM, we have seen a ESR1 methylation both in CTC and corresponding ctDNA from the same patients. And moreover, following patients over time, we have seen that ER methylation was present throughout pre-chemo, post-first treatment, and post-third treatment in endocrine therapy patients. And here you can see that this methylation was related to PFS and OS. Going on with prostate cancer, an important biomarker that I would like to focus on is ARV7. It's now known that androgen receptor has splice variants, and the splice variants that are lacking this ligand binding domain are highly important for response to therapies. It looks like it is now certified that ARV7 is highly important for selecting patients in metastatic castration-resistant prostate cancer. The patients that have that are expressing ARV7 in CTCs lack the target for abiraterone and enzalutamide, and thus respond better to taxane-based therapies. The presence of ARV7 in CTCs has been shown in many different studies using different technologies, like the one here, where the presence of nuclear protein for ARV7 is shown in CTCs. And this approach was compared with the RT-qPCR approach and published recently in a clinical trial. And in this trial, it was clearly shown that ARV7 positive patients have worse overall survival and worse PFS. The presence of ARV7 splice variants was shown as well using the Parsortix. Here you can see some nice pictures after CTCs were isolated through the Parsortix. And in this study, ARV7 was in situ detected using padlock probe technology. Moreover, another technology based on, again, on a size-based isolation of CTC and RNA-based droplet digital PCR has shown the presence of ARV7 positive CTCs in prostate cancer patients. In our lab, we have developed a multiplex RT-qPCR assay for quantifying AR splice variants in CTCs of patients. And we have seen a completely different profile in 69 patients tested. And you can see here how heterogeneous in a quantitative format this ARV7 and AR splice variants can be. We have patients that have only AR full length, or another patient that has only ARV7. This is highly important and may be critical for resistance to therapies. Concerning non-small cell lung cancer, some important biomarkers that I would like to mention are ALK rearrangements and PD-L1. Concerning ALK rearrangement that is connected with crizotinib treatments, we have to say that size-based filtration has led to beautiful studies where CTCs and were checked through FISH for ARC rearrangements. ARC rearrangements were crystal clear present in CTCs, and not only that, but CTCs that were positive for ARC rearrangements were indicative or response to treatments, to crizotinib. Patients where these CTCs were increased had a worse response, while patients where these CTCs were reduced in numbers were better responding to treatment. In this paper, which was presented last year in Corfu, sorry about that, it's in Corfu, we have to show we have seen different alterations in CTCs presented and ARC rearrangements, and these results in this paper highlight the genetic heterogeneity and clinical utility of CTCs to identify therapeutic resistant mutation in ALK rearranged patients. So beyond ALK rearrangements, a mutation in EGFR, KRAS, and BRAF genes and p53 genes were detected. This information could help clinicians for treatment personalization and resistance option to ALK-targeted therapies. Now, concerning PD-L1 in non-small cell lung cancer, all data collected up to now in numerous studies have shown a considerable heterogeneity in the PD-L1 status of CTCs from non-small cell lung cancer patients, and an increase of PD-L1 positive CTCs holds potential to predict resistance to PD-L1 and PD-1 inhibitors. This is an epitope-independent methodology based on Parsortix that was used to assess PD-L1 expression in CTCs of non-small cell cancer patients. And you can see here that in the same patient, there were CTCs that were PD-L1 negative, PD-L1 positive, and using this technology, the PD-L1 positivity was really high, 61% in the samples, while when the same samples were checked with an EpCAM-based system, the positivity was much lower, about 32%. In this nice study from the University of Crete, from the group of the University of Crete, different enrichment technologies was compared for checking immunotherapy markers in CTCs of non-small cell lung cancer patients. And it is obvious from this table that when size-based isolation of CTCs were used, the CTC-positive patients were much higher in the group of patients studied. And this study was performed after performing spiking experiments and validation of the protocols. Again, using confocal microscopy, in this study, Galatia Kalergi has shown the co-expression of PD-1 or with CKs or co-expression of PD-L1 with CKs in patients with non-small cell lung cancer. Completely using another completely different technology, this group from Paula Kajanika has shown that PD-L1 and TMT markers are co-expressed in CTCs from patients with non-small cell lung cancer under treatment with the immune checkpoint inhibitor nivolumab. And the presence of PD-L1 positive CTC seems to identify a population of patients who are not responders to nivolumab. When we compare directly CTCs and ctDNA, we have to say that ctDNA will look like a smoothie where you have a lot of information but coming from different cells, while when you have CTCs, you are actually having the real cells and you can check in depth a lot of information on them. This is a slide from Nicholas Stocklein from the University of Düsseldorf, and I like it very much and I would like to acknowledge him for this idea. Now we have performed a direct comparison study on PI3 kinase hotspot mutation in DNA isolated from EpCAM-based enrichment and paired plasma ctDNA. And you can see that in most cases, CTCs and ctDNA, these are the same blood draws. CTCs, in CTCs and ctDNA, we see exactly the same mutations. However, the concordance was much better in metastatic breast cancer than in early breast cancer. You see here that CTC and ctDNA for hotspot mutation in PI3 kinase are almost the same, but there are cases where we have this information only on CTC, or this information only on ctDNA. So this information is getting complementary. What is highly important, though, is that we have seen that in patients where no CTCs were detected according to the cell sets like here, we did detected mutations in PI3 kinase, not only in CTC, but the mutations were verified in corresponding plasma ctDNA as well. And this I can explain by the fact that detection of CTC with cell search is based on cytokeratins, while these mutations were present at a molecular level and could not be seen just by checking cytokeratins. Moreover, we had some patients like here where they had a very high number of CTCs, but the mutations were completely absent. And this is striking since we do believe that molecular characterization is highly important and we cannot rely just on CTC enumeration. And these are the percentages from DNA isolated from CTCs and before and after treatment in the metastatic breast cancer setting, in the early breast cancer setting. I would like also to show a case study where we have a patient with breast cancer that we followed since 2010. This year she died, and we are now preparing a case study paper based on this information. We have CTC isolated from this patient all over through these years, and we have performed molecular analysis at all the genes that we do, and also immunofluorescence and cell search analysis as well. You can see here the counts of CTC rising through the years, and you can see here the presence of PI3 kinase mutation, exon 20, PI3 kinase mutations again, and PI3 kinase mutations in exon 9 during the different time points. It's highly important to point out that the primary tumor was negative for PI3 kinase mutation and for ESR1 mutation and for ER methylation. And we also performed single cell analysis in these CTCs, where we have seen an increase in copy number profiles, and this we check it further in detail now. However, what is highly important in liquid biopsy analysis is quality control and assay standardization. To use CTC assays in clinical trials, it's highly important to use very standardized and validated CTC assays. And this is because when it starts a trial, you cannot change the assay by saying that now we have a better assay to use. The assay to be used throughout the trial should be very standardized and very validated so that there are no questions concerning the results coming up from CTC analysis. Based on that, I would like to point out that analytical validation and clinical validation are highly important to show clinical utility. So for CTC analysis application in clinical practice, this pyramid is highly important and should be followed. For standardization, we must have in mind pre-analytical issues, sample volume, collection tubes, and everything, analytical validation of the assay, sensitivity, specificity, robustness, repeatability. It's good for the labs to take responsibilities for analyzing CTCs for clinical trials to be ISO 15189 certified. And under all these conditions, we can move on to clinical validation and the clinical quantification for specific use. Many studies have approached the fact of standardization. These are 3 publications in clinical chemistry from our group in Cancer ID, targeting pre-analytical issues on ctDNA and gene expression in CTCs. And here you can see from our lab study on different tubes and how long gene expression can be reliable using different types of tubes for CTC analysis. Classical clinical chemistry Levy-Jennings graphs, but this time for CTC gene expression analysis. And these are highly important to follow up the stability of the assays over time. As a conclusion, I would like to say that there is no question now that liquid biopsy analysis based both on CTC and ctDNA enable us to follow cancer patients over time, enables to check for minimal residual disease, to see the effectiveness of specific drugs, but also to detect early on relapse. And CTC enumeration based on epithelial markers has already proven to be a significant prognostic marker in metastatic breast, prostate, and colorectal cancer. However, looking beyond purely epithelial CTC enables us to assess a more relevant set of CTCs, and molecular characterization of CTCs based on protein and genetic markers offers a huge potential to provide much more value to clinical trials. CTC analysis can complement ctDNA on information at the genetic and epigenetic level and has the advantages over ctDNA since beyond DNA provides information at the gene expression and protein level. Finally, working with standardized and validated CTC assays in appropriately accredited laboratories is essential for clinical trials. And some future perspectives, I think that it is now time for information derived from CTC detection and analysis to be translated for patient care. CTCs can clearly identify resistance to therapy in the metastatic setting, and there is now an opportunity for pharma to address this. And information provided by CTC molecular characterization should be used for new theranostic applications. For me, pharma should work with experienced CTC labs who have the expertise to deliver validated assays and then use these assays within clinical trials. Thank you very much for your attention, and I will be ready to answer any questions later on. Thank you.
Thank you ever so much there, Evie, and apologies again for the technical issues there. And thank you ever so much for adapting so well to those and going back and covering those important slides. That presentation was fantastic, and you really noted the importance of working with experts in accredited labs for CTC analysis. So with that in mind, I'd really like to introduce Dr. Madeline onto the stage to share her audio and visual and build upon this. Okay. Great, I can see you on the screen there. So Dr. Repellet joined Angle in 2020 as a head of clinical laboratories. She has had years in global laboratory management experience within the clinical labs in the US, the Netherlands, Belgium, and Italy. And during that time, established clinical laboratory quality management systems compliant with CLIA and ISO 15189. Okay. I'm looking forward to hearing from you, Madeline, so I'll let you carry on.
Thank you so much. Thanks for the introduction, and thanks everybody for sharing this time with us and giving me the opportunity to talk about my experience in clinical laboratories. So as you heard, my name is Madeline Repellet, and I'm head of clinical lab operations for Angle. And as a cytotechnologist, I've worked in oncology for over 34 years and in CTC testing for 21 years. So I want to thank you today, and I want to talk for the next 10 minutes or so about some of the experiences that I had on successful integration of CTCs.
Sorry, Madeline, I just want to check you're sharing, changing screen.
Yeah, I'm trying to,
okay.
And it's not working.
Okay, have you clicked on the, perhaps if you exit full screen mode and try again. Sorry about this.
Okay.
Okay, I'm going to make you presenter again and see if it works by doing that.
Okay, thank you.
No problem. Yeah, obviously I don't want the same issue to happen as last time. Okay, that should have just come through. Okay, if you change your screen, please.
How do I change the screen? What can you see right now?
I can see a full PowerPoint and yourself.
Oh, perfect. So let's see.
Great. Okay, that's working. Fantastic.
Awesome. Sorry about that. Okay,
No,
so when I studied cytology more than 30 years ago, circulating cancer cells were something that didn't seem to be very interesting for the medical field. And at the time, we were very focused on, in cytology, mostly on screening, as you know, with pap smears, etc. And so circulating cancer cells were seen as something that was a little bit too little, too late, and not very interested in exploring. And another reason was the difficulty of capturing such elusive rare cells among billions of other cells in a tube of blood. So it sounded like an unsurmountable effort. But new technologies in CTC identification have emerged, and also current drug treatments that now make it even more interesting to actually work with liquid biopsies and circulating tumor cells. In addition, in the last 10 or so years, we can see a lot more new technologies that offer a lot of opportunities to get that kind of cells that we can get information, both molecular and in complete tumor cells. Right, so during those 21 years working with clinical trials that have included CTC testing, I have learned some factors that greatly increase the chances of success integration of the CTCs into the clinical trials. And these include assay selection, which is mainly technology driven, laboratory selection, and communication. And the type of communication varies on what part of the trial you are working on. Is there's some aspects that are very important prior to start of the trial, some during the trial, and some after the trial. And we will talk about the different types of communication and the importance of each of them. Now, selecting the right assay for your study is crucial for your success. When you're interested in CTC identification, there's a few things that you should be thinking of. So, for example, what kind of CTCs are you interested in? Are you interested in just epithelial cells? Are you interested in mesenchymal cells? Are you interested in single cell counts, or are you interested in clusters? And we just heard Dr. Liandu talk about the importance of many of these types of cells. So this will help you decide both the technology that you want to work with and the laboratories that you want to work with. Things that you want to talk about too with the lab is if the assay has been fully validated and standardized. And we already also heard Dr. Liandu talk about that. Is it consistent when you run samples between different instruments, different users, different laboratories? So the robustness of the data is very important to ensure data integrity. And to enhance the use of this sample, another thing that you want to think about is, are you interested in biomarkers like PD-L1, HER, ER, and many others that are emerging lately, or even new ones that are not known yet? And I have to say, I have seen an increase in interest in biomarker testing in the last 10 years or so. And CTCs are moving away from enumeration only and going more into all kinds of characteristics, biomarkers, and molecular work, as also we heard in the previous conference. All right, so another important factor for success is obviously selection of your laboratory. And depending on your study, you may have local or global collection sites. So find out if the lab will be able to receive samples from faraway locations like Australia, Japan, South America, and other parts of the world where you may want to have clinical sites within the stability of the sample. Now, how can you be ensured that samples will not be lost due to late or weekend deliveries? So find out the hours of operation of the lab. I have seen plenty of occasions where the lab hours are not long enough to ensure that the samples can be received and processed, and particularly on Fridays, if the lab is not open on Saturdays, that really makes it very difficult to process those samples, and some of them can be lost. And this can be detrimental for global trials, and it can significantly increase the percentage of sample loss. Another thing that is interesting to know is that rejection criteria for the lab. So there are some laboratories that have certain safety requirements that do not allow, for example, HIV testing or testing of samples from HIV patients. So this is something you want to find out before you start your trial. Of course, if your exclusion criteria includes HIV, and so you will never have HIV patients, and that does not affect you. But if that is not an exclusion criteria and you will have HIV positive samples in your study, then you will have to exclude a complete part of the population from participating in that CTC testing. Another thing that you want to look into is the expertise of the management and the lab personnel, their qualification, how much do they know about working with biopharma and clinical trials? Do they understand the regulatory processes associated with clinical trials? And another important is R&D personnel. What are their qualifications and their expertise for assay development? Quality system, extremely important. Obviously, for clinical trials, they should be compliant for good clinical practices, and they should also be good clinical laboratory practice compliant. And then additionally, how do they prove this compliance by audits? You can audit them yourself, and they should always be willing to let you come into the facility and audit them and ensure that they are compliant with those regulations. But also, if they are accredited or they are looking for accreditation, for examples are ISO 15189, ISO 17025, or CLIA. If they have that, you can feel comfortable that they are being audited by external organizations periodically, and that may be a reason to limit the times when you go on audit. All right, now communication is extremely important, and we cannot overestimate the significance of communication during a study. It is key in the process and varies depending on the study stage. At the start of the study, communication must include aspects like collection and shipping instructions. This information must be very clear for the site so the sites can be trained appropriately. I have seen cases where the sites ship the samples to the wrong lab. So if the instructions are very clear and they have the right kits for the study, that is something that helps prevent that kind of errors. Sadly, the most common error I've seen is low volume. CTC testing usually requires a high volume between 7 and 10 mLs of blood, and that is not always easy to obtain. Some sites will still hope that if they send you 3 mLs of blood, you will be able to process the sample. Unfortunately, not only do they waste the time and blood of the patient, but a very expensive shipping, mostly when it's from out of the country. Shipping can be hundreds of dollars. So it is very important for the sites to be very well informed that these samples will not be processed. Do not even go ahead and ship. And then communication back to the sponsor is extremely important. It should be, the lab should be able to communicate immediately so that if possible, the samples can be redrawn within the time that is approved in the protocol. Now, talking about the data transfer agreement, this is an extremely important document that provides the right information for the lab into what kind of data is important for the sponsor. That includes the patient identifiers, a study ID, of course, the results, but there may be other information that the sponsor wants, and being very clear from the beginning will allow the laboratory to do a very good preparation of their laboratory information management system, where they will have the protocols with the right format that they can do the transfers in a monthly basis or whatever periodic basis it is, with having everything that they need there. Communication during this study is also very important, and like I said, sample issues need to be communicated right away, mostly in order to allow for either retraining of sites or for the redraw of samples. And then at the end of the study, communication is very important. I've seen a lot of issues with data cleaning and queries that were affected by lack of communication even in the middle of the study. So, for example, clinical sites had collected a sample and for some reason never shipped it, or shipped it but never sent any information to the lab, so the lab could not track it. And so the sample may have gotten lost or gone to the wrong place, and there was never any record of being received anywhere. So it's very important that the clinical sites are informed and that they communicate with the lab and let them know, I'm sending this sample today. This is the FedEx tracking number or airway bill number, and the lab will immediately track it. And if it's lost somewhere, they can communicate with the shipper and make sure that the sample is received on time, or make special arrangements to have personnel out of the regular hours to make sure the samples will not be lost. All right, and with this, I would like to introduce Angle Clinical Laboratories. We will be opening in 2021 with locations in the UK and the US. We will have 2 shifts and weekend open hours. Management with more than 30 years experience in clinical laboratory operations and more than 20 years on CTC technologies. We have a highly specialized R&D team, and the quality system will be compliant with GCP and GCLP. We are expecting that the clinical lab in the US will be CLIA licensed by Q3 2021, and both labs will be ISO 15189 accredited by also Q3 2021. We're very excited about these new labs, and I am very happy to use everything I have learned in those last 30 some years and last 21 years in clinical studies to set up our new laboratories and offer you the best services possible. So with that, I want to thank you all for listening to me and participating today, and I will be taking any questions you may have. Thank you.
Thank you very much there, Madeline. Great. I would like to now invite Evie back onto the stage. If you could share your video and audio there. Thank you. Okay, great. So thank you both very much for that presentation. It's really good to hear sort of both sides there. We've got some questions coming through. So thanks everyone who has asked questions so far. And we'll kick off with this one. Evie, how do clinical trials that last several years combat the rapid advances in CTC isolation and multi-omic characterization? By the time you've designed a study, got funding, and started recruiting patients, you are already out of date.
You mean for clinical trials?
Yeah.
Okay. Clinical trials. There are many technologies coming up every single year, and the point is that when you start a trial, you have, as very nicely Madeline said before, that it's very important to start based on assays that are very well validated and ISO 15189 validated or CLIA validated labs. So this is not changing the way that you go on with the trial. You start with the trial and you have to select in advance the correct assay to go on. And if this is validated, you can go on. You cannot stop the trial and get another design based on other technologies. So you have to be very careful from the very first moment, as very correctly Madeleine pointed out. I fully agree.
Fantastic. Okay, thank you for that one. And Madeleine, for you, how many markers can be looked at any one time on a CTC?
So how many markers will depend on how many colors you're using with your microscope. And of course, any colors that you use, so for each new color, it would be a new marker that you could use, but you would have to optimize it and validate it to make sure that there's no interference between one channel and the other, and that information you're getting is accurate.
Cool.
Can I also give an answer of that?
Of course you can. Yeah, I was about to pass over.
Madeleine replied to your question based on imaging technologies. I can give an answer on my side from the molecular assays, and I have to say that molecular assays are highly powerful because through PCR and multiplex PCR, and when I say PCR, I mean at the same time real-time PCR, but at the same time droplet digital PCR that we are using now in my lab, are highly powerful for multiplexing. And in this way, you can do at the gene expression level or at the mutation level, numerous different markers using the same material.
Okay, great.
Perfect.
Thank you for that. So,
if I can add very quickly to that, in our lab, we are working with getting 5 colors so that we can provide that amount of information.
At the protein level, which is very important. Yes.
Next question would be to both of you. Could you comment on the potential of CTCs for selecting patients for immune checkpoint drugs? Evie, perhaps if you start this one.
Yes, thank you for this question. It's very important in our time. We have already performed a study on head and neck cancer, actually using the Parsortrix, and we have shown that patients after the end of chemotherapy are that have high PD-L1 expression in the CTCs have a worse prognosis. And now there's a study under nivolumab that is selecting patients based on PD-L1 expression. So it's very important for selecting patients that are and follow up patients over time based on specific markers expressed in CTCs. I think it's a very powerful tool, CTC analysis, for this type of application for clinical trials. And I hope that Madeleine will also agree. I don't know.
Yeah, I absolutely agree. And I would just want to add something related to the regulatory aspect, and it's the fact that if you are selecting the patients based on a particular result of the study, then at least in the US, the lab has to be CLIA certified.
Yes, of course. In US is CLIA, in Europe is ISO 15189. It's the same thing, but in different continents. Yes.
Cool.
True.
Thanks for clearing that one up. And then another, you shared that CTCs with an EMT phenotype have a prognostic value in primary breast cancer. Has that been shown for other cancers, so presumably to you, Evie?
Yes, there are many studies. Unfortunately, during this half an hour, it was impossible to show all the spectrum. But I can verify this in the meeting that we have. It's 4 days meeting, and you hear all the spectrum of CTC analysis. Numerous of studies are always presented, and if you check the literature, I can give another talk only on EMT non-small cell lung cancer, EMT on different types. Yes, there are many studies showing this.
Great, okay, and then a build on that. You referred to looking at HER2 overexpression on CTCs. In the absence of a solid biopsy, is it too early to already use such information for selecting patients for trastuzumab?
Okay. There is a high discordance in many studies have shown that there is a high discordance in CTCs concerning with the primary tumor on HER2. There was a study published already in 2012 showing that patients with CTC positive for CTC receiving trastuzumab in early breast cancer did better when they were receiving trastuzumab, and in comparison with patients that did not. And then there was a trial that started. A trial started based on that, but it didn't show any good results. And I think that the problem is that the trial was not very well designed to do this. So I strongly believe that you cannot say that you will stop taking information from the primary tumor. This is the most important information, but to follow patients with HER2 positive CTCs over time is important. And I think that this should be checked in a better way now. There are trials, there are clinical trials now doing this, but
okay.
not published as yet.
Thank you. For Madeline, you will have 2 labs to support clinical trials, meaning samples will travel. How stable are CTCs over time?
And so we are working on ensuring that we have the right tubes so that we can have at least 96 hours of stability so that we can receive samples from all over the world.
Great, okay, thank you. And another question for you, Madeline, would be, it's actually for both of you, can you get ctDNA and CTCs from the same blood sample, or will you need a second blood sample?
Can I answer this first?
Yep.
Yes.
Okay, we have done this. It's our everyday practice in the lab, and I have already shown some results on same blood draws doing analysis in CTCs and ctDNA. It works perfectly well, but it depends on what type of tubes you are using to manage this. So if you use tubes that are fixing CTCs, at the same time, you have to check whether ctDNA can be analyzed reliably in these tubes.
Okay, Madeline?
Yeah, so I was going to say we are in the process of actually validating this. We have a project to work on being able to do both the ctDNA and the CTC counts, and I don't see any reasons why it should not work very well. I agree with Dr. Landu.
Great, thank you very much. So from that, I'm afraid that's all we've got time for, as it ticks up to the hour. But to both of you, Madeline and Evie, thanks ever so much for your time, and sorry about the technical issues we had at the beginning there. It was a pleasure to hear from both of you, and yeah, thank you very much for keeping us updated in the world of CTCs. And for the audience here, all unaddressed questions will be passed on to Angle. And thank you ever so much for submitting these questions. To Angle as well, thank you for hosting this fantastic webinar. And you can now, as an audience, feel free to press leave on the stage. This event has been recorded and you'll receive information on how to access this within the following work week. And please look out for an email from us. We'd love to hear from any feedback on today's webinar. If you have any follow-up questions, please feel free to email them to info@hansonwade.com and I will get back to you very soon. So on behalf of Angle and the CDX Engage team, thank you very much for your time and I hope you all have a fantastic and lovely day. Thank you again, Evie and Madeline.
Thank you.
Thank you, bye.
Bye.
Okay, you can press the